Okay, Bismillahirrahmanirrahim. Thank you so much, Dr. Wasim and his team for this workshop and for giving me a chance to present. So I will be briefly presenting genomic surveillance of SARS-CoV-2 and how NGS has played a key role in exploring the genomic diversity of SARS-CoV-2 and its variants in Pakistan. So briefly, I will discuss the current situation of COVID-19 in Pakistan and how genomic surveillance is done at the global level and also at our national level, and what facilities of NGS we have at NIH and how we are doing the genomic surveillance of SARS-CoV-2. So if we look at the number of COVID-19 cases as of yesterday, more than 217 million cases have been reported from worldwide, and the highest number of cases are reported from USA with 39 million cases. Similarly, India has also reported a high number of cases. If we look at the deaths, again, US, we see more than 654,000 deaths have been reported. And one of the important thing here is the number of deaths per million population. So if you see that the Argentina, there is more than 2,439 deaths per million population, which is quite high. Similarly, Brazil, the highest number of deaths per population have been reported. If we talk about Pakistan, Pakistan at the moment is at the 31st place in terms of the number of cases of COVID reported worldwide. And we are also, and if we talk about the deaths per million population, it's although quite low, thankfully. Another important, I think, finding here I would like to share with you all is the tests done per million population. So if we look at countries like UK, US, Argentina, and India, they are doing quite a high number of tests per million population. But on the contrary, Pakistan is quite far behind, and we are just doing around 78,000 tests per million population. So there is a need to increase the testing capacity of Pakistan. So if I compare the situation of COVID-19 in Asian countries, it's basically India that has reported the highest number of cases. We've seen what happened with the Delta variant cases in India. So Pakistan in Asia ranks at number 11. And again, there is a need for improvement in control of the disease, in controlling the spread, in vaccination, in vaccinating the population, and also increasing the testing capacity of the country. So I will very briefly go through this. So in Pakistan, as of yesterday, more than 1.1 million cases have been reported, and around 25,000 deaths have been registered. The most number of cases have been reported from Sindh and Punjab. And if you look at Islamabad, a high number of cases are reported from the capital as well. If we look at the deaths, basically the deaths have been reported mainly from the provincial capitals such as Karachi, Lahore, Peshawar, but other cities, Rawalpindi, Faisalabad, Multan, Swat, they have also reported high number of deaths. And the most susceptible, the most vulnerable age group that's been affected by COVID-19 is basically the elderly. I think this has been discussed quite a lot. So the age group of like 40 to 80, it's the most affected age group. If I talk about the testing facilities, so as of today, we have around 173 labs that are testing COVID-19 samples, and the total capacity that we have is around 77,000. And most of these labs, they are basically in Punjab and then in other provinces. If you remember that at the start of the pandemic back in February 2020, there were just very few facilities that had the capacity to test SARS-CoV-2, mainly due to biosafety challenges and availability of reagents and resources. So NIH was one of the first institute in the country that basically developed this facility for testing, and then later on, gradually these labs increased over the period of time. And as of today, we have around 173 functional labs in the country. Okay, coming to next generation sequencing and how it has played a key role in COVID-19. As you may all know that next generation sequencing has played a key role in first of all, the identification of this pathogen, SARS-CoV-2, back in November 2019 in China, Wuhan. And since then, the next generation sequencing has played an important role in designing molecular diagnostic assays and in tracking the spread of the virus and in identification of certain variants of concern and variant of interest. Also, NGS has played an important role in providing support in research and development of vaccines and therapeutics. So I don't remember any other pandemic where next generation sequencing has been used at this mass level and has played such an important role. Okay, so if we look at the global sequencing that has been done for SARS-CoV-2, so as of 27th of August 2021, more than 3 million sequences of SARS-CoV-2 have been deposited on global repositories. GISAID is basically the global initiative on sharing on influenza data. This data bank was basically initially made for influenza, but it has been very actively used for depositing data of SARS-CoV-2. Since the start of the pandemic, there have been 4 major variants of concern that have been reported from different countries, such as alpha, beta, gamma, and delta. And there are some variants of interest that have also been detected through whole genome sequencing. So if I talk about the variants of concern, basically these are just the 4: alpha, beta, gamma, and delta. If we look at alpha, which was basically detected in the United Kingdom back in September 2020, and it has certain characteristic mutations in the spike protein. And as of 27th of August, more than 1 million cases of SARS-CoV-2 of alpha variant have been deposited on GSID, and the prevalence is quite high, and I'm sure most of you know that alpha variant caused a major pandemic. Basically, after UK, it spread to all other countries, and it replaced other viral lineages. And also caused a high number of cases in Pakistan during the third wave. Then I talk about beta variant, which was first identified in South Africa back in May 2020. And similarly, gamma, which was reported in November 2020 from Brazil. So they have been also detected from several other countries, and including Pakistan. And lately, we all know that much of the talk has been about the Delta variant, which was first detected in India back in October 2020. And a high number of cases of beta variant have also been sequenced using the next generation sequencing platforms. So all these 4 variants, they have been sequenced and reported from Pakistan through next generation sequencing. In fact, National Institute of Health was the first institute in the country that did identified these variants through whole genome sequencing from Pakistan. And these sequences they have been deposited on GSID and NCBI and can be retrieved from that. Apart from the variants of concern, several variants of interest have been reported. I will not go into the details of this, but just to highlight here that ETA, which is basically the B1525 lineage, has also been reported from our country. There were just 2 cases, one from Multan and one from Peshawar. This work was carried out by University of Health Sciences in collaboration with Consortium Institute, UK. So apart from ETA, no other variant of interest has been reported from Pakistan as of today. Okay, so if we look at the distribution of these 4 variants of concern over time, you will see that the alpha variant, basically it was identified back in November, and you see that the peak cases were basically reported during March, April, May, and June. And then since July, there has been a considerable decline in the cases of alpha variant, and it is basically it has almost vanished from most of the countries, including Pakistan. Similarly, you can see beta and gamma, they were reported again during the same time period. But I will focus here on the Delta variant, which has really caused, which has basically replaced the Alpha variant and is now being detected in more than 99% of cases in Pakistan, in UK, in US, in most countries. So we see a high number of cases due to the Delta variant in our population. This is again just a representation of how these variants have behaved, have spread, or their prevalence during 2021. So if you look, the Delta variant cases are increasing, and rest of the variants, they have basically declined recently. Okay, now I will come to the genomic surveillance that is being carried out for SARS-CoV-2 in Pakistan. So a few months back, we took an initiative to establish a genomic consortium that will basically facilitate and that will strengthen our next generation sequencing capacities, particularly for SARS-CoV-2. So there are 8 different institutes that are part of this consortium and that are involved in sequencing the SARS-CoV-2. So if you see, they are from Karachi, from Lahore, and Islamabad. Okay, so as of 28th of August, around basically 968 whole genome sequences of SARS-CoV-2 have been sequenced from Pakistan. These are basically available on GSID and NCBI. And out of these, 361 sequences have been submitted by NIH. If you look at this slide, you realize that initially at the start of the pandemic, the sequencing capacity was almost was very low. In fact, it was all non-existent. But with the support of government and WHO and different stakeholders, the sequencing capacity has started to increase, and you see from March, April, the number of sequences deposited by the country, they have really increased. So in August, 412 whole genome sequences have been deposited from Pakistan, and this is based on date of submission and not on date of collection of the sample. So a look into the variant of concerns distribution. We can see that the red color shows the alpha variant, that were sequenced using the next generation sequencing. So 119 alpha variant sequences were deposited in May 2021, and in August 218 cases were reported based on whole genome sequencing. Similarly, if you see that a high number of cases of Delta variant were recently sequenced and deposited on different platforms. And just one case of the Gamma variant, which was detected in Brazil, has been sequenced, and there are only few cases of Beta sequenced from Pakistan as well. So I just made this slide to give you an idea on where we stand in terms of our genomic sequencing compared to other countries. This is for SARS-CoV-2. If you look at the number of cases, as I mentioned previously, from Pakistan, it's 968, so which is 0.08% of the total active cases, total positive cases. So basically, WHO recommends that the countries do a 5% whole genome sequencing of their active cases. But if you look apart from UK and US, most countries, they are below 1% of sequencing. So this is an area we need to focus on and we need to strengthen. So I'll quickly go through the work of whole genome sequencing that we do at NIH. We basically are using a combination of PCR-based methods and whole genome sequencing. We use variant-specific commercial kits for identification of the 4 variants, as well as some of the variant of interest. So after initial screening, we select samples for next generation sequencing. Okay, this is just to show you all how the alpha variant behaved in Pakistan. From this slide is basically from May to July, but the red bars are basically the alpha variant which were detected using a specific kit. And if you see that during May and June, the alpha variant was reported at a high prevalence, around 70%, but it's gradually started decreasing. And if you see in July, the percentage of alpha variant has significantly dropped to 20%. And if I talk about in August, this has completely been replaced by the Delta variant. So this is just the recent update of this COVID-19 variants detected during the past few weeks. If you see, the blue bar is basically showing the Delta cases, and only if you look at only just one case of alpha was reported during this time period. So basically, the Delta variant has replaced alpha variant in Pakistan. And this is done on PCR-based genotyping assay. This is not sequencing. Okay, so now coming to the next generation sequencing, what we have at NIH and how we are using the next generation sequencing platforms for SARS-CoV-2. So at NIH, we have Illumina instrument, iSeq and MySeq as well. We also have the Oxford Nanopore MinION and the Thermo Fisher Scientific GeneStudio 5. These are the next generation sequencing equipments that we have. And these are mainly dedicated for SARS-CoV-2 sequencing. We also have 10 staff for molecular biology and for bioinformatic analysis. So this is how we do just a workflow of how we do sequencing at NIH. So basically we select samples and we extract the RNA from the oropharyngeal or the nasopharyngeal swabs. Normally the samples are selected based on geographic location or having any severe disease or sign symptoms, or having travel history from abroad. So these are some of the selection criteria. We also try to select samples that have a low CT value, meaning they have high viral loads. So any sample with a CT value of less than 25 is a good candidate for whole genome sequencing. So we do use commercial kits for library preparation, and then we run that on our NGS equipment, and then then comes basically the bioinformatic parts, where the sequences are basically their quality is checked, and and sequences with poor quality or adopters, they are removed using certain softwares. And finally, once the consensus genome is made, is generated, we will look for mutation profiling using certain softwares, and then we also use online tools for signage of lineages and for phylogenetic analysis. So this is the data of NIH during May and August. So we sequenced 118 samples, and we found that 8 different lineages were prevalent in our population. If you see that we found a high number of Delta variant cases through whole genome sequencing. And this Delta AY.4, which is represented here in light brown color, it's again a sublineage of Delta. So we also found increasing cases of this sublineage of Delta in Pakistan. We also reported a few cases of Beta variant and Gamma and other lineages as well. So geographical distribution shows that most of the cases of the Delta variant were reported from Islamabad and Karachi, but other cases were reported from different geographic locations as well. We also found a high number of cases, a high number of importation of these Delta variant and Beta form and other neighboring countries. So this is another challenge for Pakistan to test and track these cases that are imported from other countries into our own country. So male to female ratio, there was not a particular difference between that. Just to go about the mutations. So if you see, I will not go into the details, but we found certain mutations, certain characteristic mutations of these variants, and as well as some new mutations, some rare mutations. So these mutations are, the number represents the mutation reported from different sequences. So among the total 118 sequences, you can see certain different changes were reported. And if you know that these mutations, especially the ones that are occurring in spike gene, they have their role in virus binding to the cells on diagnostic tools and on vaccines. So it's important that these mutations are monitored and reported in real time. Again, these are some of the mutations reported in the non-operating frame 1AB in the non-structural proteins, and we found some unique mutations. The red ones are the ones that occurred in Delta variant, and the green one that occurred in the Beta variant. So if we look at the phylogenetic analysis, we found that the Delta cases from Pakistan, which are here represented in dark blue circles, they were basically linked, or they showed high sequence homology with strains from US, UK, and Bangladesh. Similarly, Beta variant also showed some sequence homology with strains from these countries. So just to wrap up, at the moment in Pakistan, the main variant responsible for the COVID-19 cases, it's the Delta variant, and its sublineage AY4 is also increasing in number. And these variants have shown certain unique mutations and some novel rare mutations in their genome. And we also found that travelers that are returning from different countries, they serve as a key source of introduction of these variants into the country. And lastly, I believe there is a need for scaling up whole genome sequencing in Pakistan. I showed you previously that we have just done 0.08% of sequences. So we need to increase sequencing to see what other lineages are prevalent in our local population. And very lastly, these are just some of the research articles of SARS-CoV-2. Basically, this work was done at NIH using the NGS. So if anyone is interested in reading these articles, they can just access them through online. Here, I would just like to let you know about the G614 variant, the article which is on the top of your screen at the left side. This was, I think, this was one of the first variants of the SARS-CoV-2, which was basically detected back in March 2020. And due to this mutation at the 614 position of the spike, it gave the virus increase in transmissibility. So in March, after March, this variant basically it spread throughout the world, and we also found this variant through whole genome sequencing in our samples in June 2020. And we believe that if you remember the first wave of Pakistan back in June 2020, a high number of cases were reported. So we believe that that wave of the COVID-19 was mainly due to this variant. So this variant this mutation has now basically it has become a part of a backbone of all the viruses. And similarly, we also sequenced the first alpha variant B117 back in January 2021. And similarly, other variants were also reported through next generation sequencing. So this is all from my side. If you have any questions, I'll be happy to answer. Thank you.
Assalamu alaikum. Thank you, Dr. Massab. Can you please explain what is the difference between a variant of concern and variant of interest, and how we characterize them at genomic level?
Yes. So basically, just to simply answer that, variant of concerns are those lineages of SARS-CoV-2 that have established characteristics of increase in transmissibility or immune escape, or they have their role impact on viral diagnostics. So if a virus that has established any of these characteristics, it's called the variant of concern. And variant of interest are basically the lineages that have the potential, or that may cause increase in transmissibility or immune escape, or may affect the viral molecular or diagnostic assays. So these are basically characterized on this criteria, and these are again characterized on the basis of certain at the genomic level, certain mutations that may give the virus any selective advantage, whether it's in replication or in avoiding immune escape. I hope I answer your question.
Thank you.
Friday. Gee, thank you so much. Wonderful talk, Dr. Massab.
Thank you, sir. Thank you much.
Sir. My just probably a childish question. Why don't we use Sanger for this? Because NGS, what happened to us was we were going for big data sets, high throughput data sets. and all of a sudden COVID showed from nowhere and we realized like हमारे पास मशीनें बहुत बड़ी हैं, तो जीनोम तो बहुत छोटा है. तो how would you compare के हम इसको Sanger पे क्यों नहीं करते बजाय इसके कि हम NGS पे करें? And secondly, you need to have the, अगर आपने एक मशीन चलानी है जैसे MySeq चलानी है तो उसका थ्रूपुट है 16 GB एंड योर जीनोम जो हमारा कोविड का है ये तो
30 KB
में. तो थोड़ा सा मुझे फील हो रहा था कि जैसे हम ये इस पे हम Sanger से भी कर सकते हैं. इज नॉट इट सो?
डॉक्टर साहब बिल्कुल आप ठीक कह रहे हैं. Sanger से इनिशियली हुआ भी है इसका होल जीनोम, लेकिन अगेन थ्रूपुट बहुत इंपॉर्टेंट है. Sanger से वो थ्रूपुट में नहीं मिल सकती जो हम अभी NGS दूसरे सिस्टम यूज कर रहे हैं. अगर आप ग्लोबली देखें तो कंट्रीज लाइक UK और US वो एक बैच में यूजिंग HiSeq और NovaSeq वो 500-600 होल जीनोम्स कर रहे हैं वायरस के. जबकि अगर आप Sanger पे यही काम करेंगे तो आपको सालों नहीं तो एटलीस्ट महीने लग जाएंगे और वो लेबोरियस बहुत हो जाएगा.
उससे पहले हमें जब हम एक्सट्रैक्ट करेंगे इसको तो हाउ वुड वी इंश्योर यानी वो किट्स वगैरह के जरिए हमने इंश्योर करना है कि ओनली वायरस जीनोम इज प्रोसेस्ड इन द मशीन, राइट?
डॉक्टर साहब इसमें ये है कि दो बेसिकली टेक्निक्स हैं. एक तो आपकी मेटाजेनोमिक बेस है और एक आपकी एम्प्लिकॉन बेस है. हम बल्कि मोस्टली पाकिस्तान में भी और दुनिया में भी ज़्यादातर एम्प्लिकॉन बेस टेक्नोलॉजी यूज हो रही है. तो उस जिसमें SARS-CoV-2 के स्पेसिफिक प्राइमर्स डिजाइन होते हैं और आप जो भी न्यूक्लिक एसिड एक्सट्रैक्ट करते हैं वो डेफिनेटली टारगेट करता है स्पेसिफिक आपके पैथोजन को और उसके.
और आप ये मिनियन भी यूज कर रहे हैं अपने, किसी ने मुझे बताया था आप लोगों के पास, वो स्टूडेंट्स थे मेरे पिछले कोर्स में NGS में फ्रॉम योर NIH एंड दे सेड वी हैव दिस मिनियन USB वाला जो है, अभी मैंने शुरू में लेक्चर में भी बताया था. आई थिंक इस पे कर रहे हैं आप लोग प्रोसेस आपके पास?
डॉक्टर साहब ये इनिशियली हमने इसको यूज किया था. अब ज्यादातर तो हम अपनी इल्यूमिना iSeq और थर्मो फिशर जीन स्टूडियो S5 पे काम कर रहे हैं. मिनियन पे हमें कुछ डिफिकल्टीज आई थी. जो हमारा रिएजेंट आया था फ्रॉम अब्रॉड वो समहाउ वो ड्यूरिंग ट्रांसपोर्ट उसकी कोई टेम्परेचर इश्यूज आ गए थे. तो अब इसके रिएजेंट्स का बड़ा प्रॉब्लम है इंपोर्ट करवाने का. वो हम वेट कर रहे हैं. मिनियन पे जस्ट फ्यू सैंपल्स हमने इनिशियली होल जीनोम किए थे, लेकिन अब हम मोस्टली इल्यूमिना और थर्मो फिशर इंस्ट्रूमेंट यूज कर रहे हैं.
थैंक यू सो मच.